Hi, my name is Mike Bollong, and I am an assistant professor in the Department of Chemistry at Scripps Research. And today I am delighted to take you through two of the preclinical drug development programs that my laboratory is working on in collaboration with Caliber. And so before we dive in, I thought I'd tell you a little bit about our strategy and our research interests. So we are a chemical biology group. We make full use of the screening center at Caliber to understand if a drug like small molecule might be identified for a specific biological question that we are asking. So we use these pharmacological screens as a surrogate for doing genetics type screens. This ultimately allows us to gain new insights about cell biology, but also turns our discoveries rather quickly into the starting points really for new medicine. One particular area of interest that I think has been ill-addressed by the field generally is the area of regenerative medicine, and one that I was attracted to given its inherent challenges. So specifically, my group is interested in understanding if we might be able to induce organ repair as a general research paradigm. Our bodies are continually renewing our organs on a day-to-day basis through cell proliferation, meaning that our approach would necessarily have to tell our organs new ways and instructing them of how to induce cell division through these new pathways. At the heart of the two drug development programs that I'll tell you about today is that the idea that we might be able to instruct our bodies to repair themselves by turning on specific and appropriate proliferative responses in the context of our disease. Clearly, our bodies have an untapped capacity for regenerative potential. We just need to learn how to access that capacity using the tools of chemistry. And so the first story I thought I would tell you about today has to do with stem cells of the lung. All of our organs really contain stem cells, and our lung is no different. So as you likely know, the primary unit of gas exchange in the lower airway is a sac-like structure called an alveolus. And for the purposes of today's talk, I want you to remember one cell type in particular, and that is called an AEC2 cell or an alveolar type 2 cell. These cells secrete surfactant, which is essentially a soap-like substance so that the lung does not collapse when you breathe. But they're also the stem cells of the lower airway. They slowly give rise to AEC1 cells, which are required and necessary for gas exchange, and they promote barrier function. So why are these cells important? Well, it turns out that in addition to normal lung physiology, the AEC2 stem cell population is critically important to a disease called idiopathic pulmonary fibrosis, or IPF. So IPF had historically been largely thought of as a disease of scarring, and its source had been enigmatic until recently, at which point pioneering work by the work of Paul Noble and others has shown that IPF is really a disease driven by ineffective lower airway regeneration. At the heart of this disease is a stem cell population which is ineffective at lower airway repair. This results in two phenomena happening, one of which is the excess deposition of scar tissue, which is often irreversible and the most salient feature of the disease. But there's also ineffective barrier function, which means that other cells of the upper airway, namely epithelial cells from the respiratory tract, come in and proliferate inappropriately. This results in ineffective gas exchange and ultimately death. This ultimately led us to the idea that we might be able to find a drug that could restore this appropriate proliferative response of AEC2 cells and treat the disease at its root cause. In addition, this type of therapy might likely be additive or synergistic in terms of efficacy with the approved drugs, which only really target the extracellular matrix deposition that's observed in fibrosis. So that led us on a hunt to find a drug molecule that might do this. Now, it's often said that the best place to start for a new drug is an old one. So we asked the question, is there some existing drug mechanism that will make this stem cell population grow? So we surveyed the comprehensive drug repurposing library Reframe, which has nearly every single small molecule drug which has been tested in phase 1 clinical trials in human beings, ultimately looking for a drug that might make these cells grow or proliferate. We did a high content imaging screen with aged human AEC2s, taking many quantitative images per treatment to see whether or not these cells are growing in response to each drug treatment. We ultimately identified one series of very widely used drugs that very efficaciously activate this proliferative response in these cells, which was quite a surprise to us. We ultimately found out that this drug class modulates the innate ability of AEC2s that that ACE2s basically tell themselves how to grow. It modulates that distinct capacity. And what was really unexpected and incredibly fascinating to us, and this was fascinating from an academic perspective, it really wasn't until we observed what happened in the mouse that we began to perceive this as a potential therapeutic candidate. So what we can do with in our animal models is challenge animals with an acute inflammatory damaging stimulus called LPS, which induces dramatic lung injury. I don't think you need to be a histological expert to see that treatment with these drugs really induces robust protection effects, as you can see between the controls here. Importantly, we can actually show by histology that protection in this model is driven by induced proliferation, meaning the drug is telling the appropriate cells to grow and to repair damage, ultimately on a 5-day timescale, which is quite impressive. We additionally demonstrated that this series of approved drugs was efficacious in a different model of lung damage. In this case, a model which mimics what naturally occurs in idiopathic pulmonary fibrosis. Here I'm showing that this drug class we ultimately identified as more efficacious than current standards of care drugs. In this case, it's a drug called pirfenidone, and that's read out by multiple metrics of lung and organismal health. And so something that I'm not showing that is equally, if not more impressive, is that this drug class can be co-dosed with another standard of care drug called nintedanib, and that the combination of both drugs together results in a synergistic effect on lung health in this model that is considerably better than either drug alone. An important issue here that I'm showing in this lower right-hand corner is that the doses required for drug are incredibly high, 5 to 20-fold times that's clinically achievable in human beings, meaning that we don't think that we can achieve that dose with any of the particular drugs that are approved for this class, because they were not designed really for long-term lung exposure. And so that's not great from a direct drug repurposing point of view, but it gave us the unique opportunity to make really fantastic lung localized versions of these drugs. So these would be molecules that would be inhaled, but are chemically modified in such a way that they have long-term residence time in the lung, but do not have good exposure to the systemic circulation. Literally, the first molecule we made as a proof of concept of this idea, a molecule called NZ97, demonstrated tremendous lung partitioning when dosed to the lung. We can see from the PK curves on the right that NZ97 displays an essentially flat PK curve with high lung exposure and low plasma levels, while the approved drug it was based on is rapidly cleared within a few hours. We can additionally dose this drug at very low levels and observe protection in an acute lung injury model. This basically allows us to spare the dose of the clinically approved drug by about 50-fold. We can also observe protection with this model, basically with this proof of concept molecule and mouse bleomycin models. In this case, the drug is cleared so slowly that we can dose the drug every 4 days and maybe even once a week and still observe efficacy in this dosing regime. This has led to a very expansive drug development effort where we have explored a lot of the lung retention chemistries and tolerabilities of these compounds. And the good news is that these compounds are very efficacious and incredibly well tolerated. Our goal is to have a nebulized formulation of our preclinical candidate by the middle of this year. And ultimately, our goal is to have finished our IND enabling studies by the end of the year and be in phase 1 clinical trials at some point during 2022. So I thought I would recap some key points of this project, namely that this approach is, to our knowledge, the only regenerative approach towards treating IPF, and certainly the one that addresses IPF at its etiological core. This is really in contrast to inhibiting inflammation or inhibiting fibroblast signaling like all of the IPF molecules in the pipelines of all major pharma companies. We additionally think that these drugs will be safe because this mechanism really only augments an endogenous pathway that AEC2s themselves use to modulate their proliferative activities in response to damage. And lastly, I think it's worth mentioning that we're exploring how we might modulate lower airway repair as therapy for other disease states, including COVID-19, which is a key worry at present, COPD, which kills roughly 5% of all human beings on Earth every year, and ARDS, an indication for which there are still no good therapies. And so next, in the next story, I thought I would tell you about our work in organ size control and manipulating a key growth pathway. And so if you've ever wondered why your organs stay the same size as they do, is because there's, of course, a pathway that controls that process. In animals, organ size is controlled by the hippo-YAP pathway, which consists of YAP, which is essentially a transcriptional gas pedal telling cells to grow until organs get to a certain size and density, as well as the hippo pathway, which acts to counteract YAP and inhibit its pro-growth activities in cells. So when you delete hippo pathway components in animals, you get organ size expansion. And you can see that this pathway is well conserved from flies to mammals, as deletion of hippo in either fly eyes or mouse livers results in extreme overgrowth. So really the question is, why are we interested in this pathway? Well, it turns out of the pathways controlling mammalian regeneration, YAP is one of the very few that's actually known to be conserved in mammals. So it's known that YAP actually controls the proliferation and stemness of multiple epithelial precursor populations. So based on this, we came up with the fairly simple idea that you might be able to turn YAP on with a small molecule drug in the context of disease. This would result in reparative proliferation, at which point we could stop dosing the drug, and that would in principle mitigate specific concerns with inducing proliferative disease, namely cancer. So ultimately, our research as to good indications led us to chronic wounds as the first indication for using a YAP activating small molecule. I'm sure many of us have had firsthand experiences with chronic wounds, probably knowing someone who has had one. As an example, it's worth mentioning that there are at present up to 4 million diabetic foot ulcers at present in the United States. Clearly, this is an unmet medical need. A very shocking statistic is actually half of the people that are diagnosed with a DFU will not survive the next 5 years. Unfortunately, there's only one regenerative therapy approved. It was developed over 20 years ago, and it doesn't work very well. And here our approach is really quite a simple one. Can we identify a metabolically soft drug that activates YAP when it's applied topically? This would overcome the stalled cell proliferation that's observed in chronic wounds and help to close the wound as soon as possible by increasing cell division. Importantly, YAP really augments multiple phases of wound healing, including telling keratinocytes to grow, activating wound healing myofibroblasts, and reactivating the wound. So we think a small molecule YAP activator would be beneficial for chronic wounds, but one really didn't exist when we started this project. So we were able to ultimately identify a chemical scaffold with the requisite properties for the indication I described. So from a screen of about 800,000 molecules, we identified one very efficacious chemical series, which is idealized by our lead scaffold called PY60. This molecule activates YAP very efficaciously in essentially any cell condition. It promotes a very specific transcriptional signature telling cells to grow and to proliferate and to survive. And importantly, this molecule does not inhibit the upstream kinases, MSD1 or 2 or LATS1 and 2, which have many other roles in cells. So unfortunately, I don't have time to go into the molecular details of how this molecule works, but it turns out this molecule does target a protein which we determined to be a new member of the hippo pathway, which is a part of the density sensing machinery of the cell. So this drug basically tricks the cell into thinking it's an environment that's sparser than it is, which allows cells to keep growing and ultimately to repair the organ. This molecule should tell cells to grow, and it certainly does. We can take preparations of mouse epidermal keratinocytes and expand them essentially ad infinitum. Normally these cells would not survive, they would die because they're not receiving the mitogenic cues. But in the context of drug treatment, we can see that the stem cell populations of these cells expand, and the cells are able to survive over long periods of time. This proliferative effect is ultimately seen in whole organisms and is not just confined to the tissue culture dish. We can apply this drug topically to the dorsal skin of the adult mouse, and what we observe is pretty astonishing. Basically, within 1 week of treatment, we can see a doubling of the thickness of the epidermal layer, which is consistent with what we need in the context of wound healing. Our genetics work told us that YAP is essential for this process, and we knew that this process was dependent on proliferation because we can actually see the cells dividing in response to drug treatment. The major takeaway of these slides is to show you that this drug candidate can completely remodel the epidermis of the adult animal in response to topical treatment of drug, which would lead us to think that we can accelerate wound healing in adult organisms. So all of the work I've shown you before had been done in mice, but what about higher species? So mouse skin is actually quite a bit different than human skin, both in terms of its composition and its morphology. A much better molecule is actually the pig or the mini pig. And so here we were actually able to identify a gel-based formulation of the drug, which robustly expands the epidermal keratinocyte layer of mini pigs, roughly doubling that thickness, but it also influences the dermal layer where it's able to induce dermal hyperplasia. We ultimately think that this drug will be efficacious in wound healing because it addresses multiple phases of wound healing, basically promoting epidermal hyperplasia as well as dermal hyperplasia, which are essential for effective wound healing. And lastly, the next question, of course, is to understand if this agent might be efficacious in human patients and in the context of human wounds. Unfortunately, I can't test this agent on myself just yet, but we can test this agent on human skin by using human skin equivalents, which are reconstituted human skin grown in a tissue culture dish. Luckily, our molecule is incredibly efficacious at promoting wound closure in this setting. It promotes cell growth and wound closure essentially as fast or faster than any of the positive controls we've evaluated. And the effect here is quite striking. You can see the red dots in this histological slide are those that are induced to proliferate, and our small molecule does that quite efficaciously. So we found these studies to be incredibly compelling, and based on them, we are at the time of recording this video actually performing a number of wound healing studies in the mini pig as a next step towards declaring this molecule as an IND candidate, hopefully which we will complete by the end of this year. And so just to recap, I've shown you our regenerative approach towards treating diabetic foot ulcers, one that we hope can be added on to existing standards of care, which are implanted matrix devices along with wound debridement and offloading. We think this approach is innovative because it's the first essentially full activity YAP activator that's been used either clinically or preclinically. And lastly, I'd like to just point out that we are expanding our efforts in this space. We now have multiple scaffolds which activate YAP very potently in cells, and we're ultimately trying to generate second generation molecules, hopefully that we can take into other indications in internal organs, especially heart failure. And so we think the future of these sorts of therapies is incredibly bright, and I'm looking forward to a future where regenerative therapies are part of the standard toolkit for treating diseases of aging. I hope the work I showed you today helps to spur on efforts in this space. And now, just lastly, I'd like to thank the folks who were involved in this effort, the principal scientists leading the actual experimental efforts. Sita Shao, for example, has been running the entire AAC2 preclinical program, and Anita Dreslak and Maya Boulos have been leading our efforts into YAP. I'd also like to thank you very much for your attention, and I'd be delighted to take any questions during the Q&A or during the breakout room session. Thank you very much.